So तुमच्या NCERT मध्ये you will find two chapters separate, principles of biotechnology and second is applications of biotechnology. So in principles of biotechnology we will learn what exactly biotechnology is. Okay, how different methods are used to find or to manipulate genetic material. Okay, and in applications we will see how using biotechnology we have already developed some organisms with new genetic makeup. We have treated some of the diseases. We have made some organisms or plants or animal varieties having new combinations. Okay, ते सगळा पार्ट जो आहे तो applications मध्ये. Okay, so first part is principles of biotechnology. What biotechnology exactly is. Okay, so bio is life, technology are techniques. Okay, here we are using techniques to develop or to change living organisms. Okay, आणि त्याचाच एक पार्ट आहे रिकॉम्बिनंट डीएनए टेक्नॉलॉजी. नॅचरली रिकॉम्बिनेशन टेक्स प्लेस. ओके, कुठे होतं रिकॉम्बिनेशन नॅचरली? Meiosis. What is recombination? New combinations of genes, right? That is recombination. So in recombination, some genes of one chromosome combine with some genes of other chromosome आणि त्यामुळे एक नवीन जेनेटिक मेकअप तयार होतो which gives unique characteristic feature to resulting zygote, right? So naturally recombination takes place in meiosis, but in recombination genes which are present on two non-sister chromatids get exchanged. Okay, here we are talking about genes which are never present in that species, never going to develop in that species, and we are thinking of introducing those genes in these organisms. Okay, for example, um, सगळ्यात पहिलं रिकॉम्बिनंट डीएनएचं काय एक्झाम्पल आपण नेहमी जे स्टडी करतो ते की सी इन्सुलिन डिपेंडंट डायबिटीजमध्ये बिकॉज ऑफ डेफिशियन्सी ऑफ इन्सुलिन glucose level increases, and now these patients are supplied with insulin injection so that their blood glucose level will remain normal, right? Okay, from where this insulin comes? Okay, so this insulin, earlier days, this insulin people used to extract from pancreas of either dogs or horse. Okay, and that insulin, which is similar, functionally it is similar to human insulin, but chemically it is slightly different than human insulin. Okay, so even if in those organisms insulin brings down blood glucose level, the chemical combination or amino acid sequence which is present in those insulin and that of human insulin is slightly different, and therefore some people may get allergy to this unknown new insulin. Okay, second thing, that insulin extraction from pancreas of these organisms is quite expensive. Okay, and we will get hardly some quantity of insulin, and that insulin will be expensive. But now number of diabetic patients are increasing, and therefore we want insulin supply in cheaper uh, availability, and that too exactly human insulin. So what scientists have done, they have taken gene of human insulin from humans. How? We will study. Okay, but त्यांनी केलं काय? They have cut down the gene which codes for human insulin from human genome, and that gene they introduced in E. coli, which is a bacteria. So bacterium doesn't require insulin at all because it has no place to store insulin, to convert glucose to glycogen and store in cell. It is never going to evolve such that it will produce insulin and it will start storing glucose in the form of glycogen. Okay, but artificially we have introduced that gene in the genome of E. coli. E. coli cells do not know that they have this additional gene in them. Okay, and they are transcribing this gene of insulin. They are translating this mRNA, and they are producing large quantity of insulin for what they don't know. Okay, and this insulin we are extracting from these microorganisms, and we are putting it in bottles and selling it market. Okay, it's very easy to grow these microorganisms of bacteria in laboratory. So in small laboratory large quantity of bacteria can be grown. Once this insulin gene is introduced in one E. coli cell, all its daughter cells will retain same gene. Okay, so जेव्हा one E. coli will become two, it will produce two daughter cells. Both the cells will have gene for insulin. Okay, further generation will also have gene for insulin. Okay, so this is how many hormones, many enzymes, many factors are produced by microorganisms, okay, by introducing this gene. Sometimes we want to introduce some useful gene in plants also. So in plants also, this is how we can introduce any gene. For example, a gene which produces vitamin A. We have studied that golden rice is good supplement of vitamin A. So biotechnologically, vitamin A producing gene is introduced in rice, and that rice is now rich source of vitamin A. So along with carbohydrate, it is producing vitamin. Okay, so this is how gene from one organism is extracted and introduced in other organism, that is called as recombinant DNA technology. Okay, now how to cut down and how to extract that gene we will see. How to insert that gene in bacterium we will study. Okay, so what are steps involved in this recombinant technology we will see. But once you know that you can extract any gene and you can introduce it in any organism, you can decide what combinations you want, what result you will get, okay, and accordingly you can manipulate all this genetic makeup. Okay, this is recombinant DNA technology, and this is all about biotechnology. Okay, so in biotechnology, we are using some microorganisms, some enzymes from microorganisms, some genes from various living organisms. Various living organisms, not only bacteria, we are using viruses, we are using plasmids. Do you remember what is plasmid?
ते डीएनएचे हे असतात ना टॅक्स विदाउट सेलमध्येच असतात
कोणत्या प्रोकॅरियोटिक
युकॅरियोट प्रोकॅरियोट
both A and B, none of these. Option A prokaryotic, option B eukaryotic. C both A and B, D none of these. हाँ एकाने सांगितलंय A, एकाने सांगितलंय B. रुद्र you would say C. हाँ यश will say D. काहीतरी कोणाचं तरी येईल बरोबर.
युकॅरियोट वाचलेलं आठवतंय मला पण आठवतंय तो लेफ्ट साइडचा पॅराग्राफ आहे
It is an none of these. Okay, you are saying none of these. Yash?
आय गेस ओके
पहिल्यांदाच आहे यश. हा पहिल्यांदाच ऐकलं प्लाझमिड. Now they both are saying eukaryotes. शोधायचं नाही इकडे तिकडे. Okay, it is present in prokaryotes. Okay, so prokaryotes have extra chromosomal DNA along with their nucleoid. They have one additional small DNA molecule. Okay, and this small DNA molecule codes for some genes. काहीतरी genes त्याच्यावरती आहे. Okay, and some bacteria do not have plasmid at all. So genomic DNA has to be there. Okay, then only cell will be live. Okay, without genetic material, cell will not survive. But plasmid can be there or may not be because it is very small DNA. Okay, so that is optional. If plasmid is there, that is an add-on property. If plasmid is not there, it doesn't matter. Bacteria will survive. Okay.
प्लाझ्मिड न्यूक्लियसच्या बाहेर असलेला डीएनए
न्यूक्लियसच नसतो ना बॅक्टेरियामध्ये.
प्रोकॅरियोट
ओके, सो बॅक्टेरियामध्ये न्यूक्लियस म्हणजे नाहीच आहे. सो मुळात त्याचं जेनेटिक मटेरियल इज ऑल्सो इन सायटोप्लाझम. So plasmid is also in cytoplasm. Okay, so this additional small DNA which is present in bacteria, okay, the bacteria को उसका जितना यूज नहीं है ना उतना यूज हम कर रहे हैं. Okay, how we will see. Okay, so आपण काय म्हटलं, recombinant DNA is what? Take out gene from one organism and introduce it in other. Okay, but how to take out? Okay, how to take out exactly the gene and not previous and later gene. बाकीचा कचरा आपल्याला नको आहे. We want exactly gene which codes for human insulin. How will we know that it is present on first, second, third? Out of 23 chromosomes, it can be anywhere. Okay, so that exactly we have to identify and take out. Okay, and all this is chemical. Okay, you are not going to look at entire DNA. This is the sequence. This is the gene of insulin. Caesar लेके उसको microscope के नीचे लेके cut करो. It is all chemical. Okay, so theoretically you know what to do, and all the processes you are doing is in cuvette or say Eppendorf tube or small test tubes. Okay, so in test tube you are performing some chemical reactions, and you have to believe that whatever the precipitate is DNA. Okay, then you have to cut that DNA in small fragments. You have to separate all fragments. Okay, and then from separated fragments you have to take out exactly gene coding for insulin. Okay, which you cannot read by eyes. Okay, सो ये निकालना है, उसको इंसर्ट करना है दूसरे ऑर्गेनिज्म में, दूसरा ऑर्गेनिज्म विल स्टार्ट यूजिंग दैट जीन, देन इट विल प्रोड्यूस द प्रोटीन, वो प्रोटीन एक्सट्रैक्ट करके एनालाइज करने के बाद यू विल अंडरस्टैंड वेदर इट इज इंसुलिन और नॉट. If it is insulin, your process was correct. Okay, if it is not, again start with extraction of DNA. Okay, so this is how recombinant DNA is done, and this is how various chemicals are used for all the processes. Okay, because we are not going to actually see the gene, the transcription, the translation, mRNA, protein, we cannot see anything. Okay, but by chemical reactions we will do all these steps. Okay, so all some steps are given here. Okay, now biotechnologically we can artificially amplify DNA also. Okay, and that means during corona pandemic, they used to say कि RT-PCR test करेंगे. Okay, whenever they used to take corona samples, okay, from nose, they used to carry out RT-PCR. Okay, so what is that PCR? That also we will study in this chapter. Okay, PCR is polymerase chain reaction. So what exactly PCR is and what is RT-PCR? All that we will study in this chapter. Okay, so the chapter is most interesting chapter, most advanced chapter as compared to entire 11th and 12th syllabus. Okay, and if you understand this chapter, this biotechnology has a bright future, because using biotechnology you can enter in medical sciences, you can enter in environmental, you can enter in any kind of biology related field. Okay, because biotechnology is the coming branch. Okay, so it is let's say amalgamation of engineering and medical. Okay, so medicine और engineering का ये fusion है जहाँ पे you are using technology also, you are using living organisms also. Okay, चलो let's start with this. Now the sequence given in your textbook is थोड़ा सा shuffle करावा लागेल आपल्याला बघू कसं काय तुम्हाला सवय लागते. ओके, तेच बुक ठेवूया, आपण एकच बुक फॉलो करूया. एनसीईआरटी मध्ये दोन चॅप्टर्स आहेत, एक आहे ऍप्लिकेशन प्रिन्सिपल्स ऑफ बायोटेक्नॉलॉजी आणि दुसरा ऍप्लिकेशन.
मी लाईक बारावीच्या सिलेबससाठी घ्यायचं
हो. Okay, you are already aware of what biotechnology is. It is product of interaction between biological science and technology. It is in fact an applied branch of biology. Why I have said you are already aware of, because in 10th you had a chapter called biotechnology. Okay, so here in biotechnology you have studied की काहीतरी आपल्याला engineering tools, techniques वापरायचे आहेत आणि living organisms. The term biotechnology was first used by Carl Erecki in 1919 to describe process of large scale production of pigs. Okay, so काहीतरी artificial केलं म्हणून त्याने म्हटलं biotechnology. It is defined by different organizations in different way. It has been broadly defined as the development and utilization of biological forms, products, or processes for obtaining maximum benefits to man and other forms of life. Okay, we are so selfish. Okay, we are using microorganisms, we are using their enzymes, we are using normal processes. Processes means transcription, translation, okay, natural recombination, okay, replication of DNA. All these are biological processes which happen in cell, and these processes we will manipulate the way we want. Okay, so process म्हणजे त्यांना हे म्हणायचं आहे. Okay, replication, transcription, translation, recombination, all these processes. According to OECD, that is Organization of Economic Cooperation and Development, it is application of scientific and engineering principles to the processing of material by biological agents to provide goods and services to human welfare. It uses scientific principles of microbiology. What is microbiology? Study of microorganisms, that is bacteria, viruses, unicellular plants, fungi, okay, and small animals also. Okay. Genetic biochemistry means transcription, translation, all the enzymes involved, replication. Chemical engineering, okay, all the chemicals which are used to carry out various processes. Mathematics, because there is statistical analysis, so mathematics म्हणजे फार काही कॉम्प्लेक्स नाही आहे. Okay, because many times biotech engineering म्हणजे engineering म्हणूनच लोक बॅकआउट करतात. Okay, but in biotech engineering there is 10% engineering means mathematics and 90% is biology. ओके, सो इथे मॅथमॅटिक्स कुठे लागणार आहे? फॉर स्टॅटिस्टिकल अनालिसिस ऑफ डेटा. ओके, स्टॅटिस्टिक्स, computers, industrial processes. Why we need computers? Because last time we have said what is genomic and proteomics. Genomics means what? It is the genome sequence of each organism, and this data is processed and it is stored on computers. So that is called as biomics. Okay, it is genomics and proteomics. Okay, so त्याच्यासाठी आपल्याला लागणार आहे computers. Industrial processes, because there is large scale production of various vitamins and chemicals. Biological agent means plants and animal cells, microorganisms, enzymes are their products. Okay. History of origin of biotechnology is as old as human civilization. Development of biotechnology in terms of growth occurred in two phases, that is traditional biotechnology and modern biotechnology. So in traditional biotechnology ते काय large scale curd making आणि bread making त्याला ते biotech म्हणायचे. आधी तर pigचं production, that is biotechnology. But in modern biotechnology now we are more focused on recombinant DNA. Okay. So traditional biotechnology, it was primarily based on the fermentation technology using microorganisms in the preparation of curd, ghee, soma, vinegar, yogurt, cheese making, wine making, etc. It became an art of kitchen in Indian houses. Okay, so it is not a technology; it is more like a art. Okay, if you know exact how much inoculant or microorganism is to be added in milk, then you will get exact proper taste of that curd. Okay, so that is art. It is not technique. Okay, it was more of art than science. Till that time, people did not know about how exactly the process occurs and organisms causing the process. The